Thank you very much. Bartolome and Beatman, a fantastic duo, as you've heard. Everything new, full of energy, and you can feel the fun that they're having in composing and executing this. Buy their records, go on Spotify. Bartolome is Matthias Bartolome on cello, and it's not just a cello, but it is L'Éclair, which is also the name of the song, because this is a cello from 1727, done in Rome by probably the best Baroque cello maker at that time, David Tekler. And he's accompanied, or his accompanied is different to say, and his partner plays a violin that has grown to be jealous of the Tekler, and so is always keen that he's mentioned, and is made a little bit later, only in 1817, actually from Josephus Pauli in Linz. And this is played by Clemens Bittmann, who is the partner of Matthias Barthelemy. Thank you so much. We're looking forward for your next songs after the introduction. So thank you very much. So, ladies and gentlemen, welcome to the 16th CeMM Karl Landsteiner lecture. As you know, this is for us is the event of the year. We always say this is the gathering of the tribes, and indeed it is the gathering of the tribes. A lot of our hardcore stakeholders and supporters are here, and I welcome them all in a global sense. We also have new friends, old friends, and we have the big delegation of CeMM alumni that are visiting us, which is fantastic from all over the world, because today we had our alumni meeting at CeMM. So I particularly welcome, of course, our speaker, the 16th lecturer, Benjamin Cravatt, and I'll introduce him in a second. But before, I'll show you a little bit what the here you can see again the names of the musicians, and they have a beautiful Spotify account, so tonight you can hear the song again and dance to it, because I think it's actually a dance song. Okay, so after my mini introduction, we will have the award of the Denise Barlow Award. Then I will have a very brief laudatio of Ben Cravatt. Then we'll hear his lecture. Then there is going to be time for question and answer. And very importantly, we will then have, there's not going to be many closing remarks, but there's going to be an invitation to come downstairs where we have the traditional party that is actually the networking event that allows us all to meet the speaker and to maybe meet the musician if they're still here and have fun. So this is a particularly nice event because we are back where we belong, which is this beautiful festive hall of the Academy of Science. And the Academy of Science is our mother organization, and we are very, very proud that our mother organization has this beautiful building with this beautiful festive hall. The building is done around the mid of the 18th century, 1757 or something like this. It was meant to be a new building for then the university. The university was not new because, of course, it was 300 years older than that. But at that time, it appeared important to have a new program that took away from the rather medieval rooms where the university was housed until then, and this was spectacular. And it was really the wish of Maria Theresia and Franz Stephan, and you see them in the medallion on the ceiling in the center. In the sun, this is the Empress Maria Theresia with her husband, and it was their wish also to somehow bring a little bit the spirit of Illuminism and sort of to this place. And you have the four faculties at that time that were important. You have medicine on that side. If you want to turn your head, you will have pieces of flags coming off the table. Behind me, you have philosophy, and you have both theology and you have law as the four major faculty of that time. After about 100 years, Emperor Ferdinand I, that allowed to make the academy in 1848, 1847, allowed the academy to take over this building. And it has been since then here, something that makes us happy because we can have our regular meetings here, and next week we have such a meeting. So very briefly, I would like to tell you a little bit, so welcome here, and for those of you who haven't been here, enjoy the beautiful thing. A very few things about CeMM. This is the Center for Molecular Medicine. We decided that we always say something about it. We have 5 principles that we are following. The first one is that we try to always be innovative on the technology side, and we'll hear from our lecturer today how technology is really what he has pioneered, and it's not a coincidence that he's a hero for us, because we think that without a new instrumentation, you cannot see new things. Without a new technology, you cannot make discoveries. And I think some of the frescoes here inspiring tell the same story. The other point is that we're interested at the roots of disease, so we are really interested in how does disease generate at a molecular level. So, of course, we're interested in also applying it, which is the third point, but we are interested fundamentally in understanding how it comes about. Very important for us is training, so empowering talents in all possible ways, not only the students, but also the postdoc, the technicians, the administration, and of course the principal investigators, so that through being associated with us, they are empowered and do great things. And very important, we share. So we share, this lecture maybe is an example, but we share by trying to publish in open access journals. We try to share our data. We try to make our data shareable to start with, and so on and so forth. So very, very briefly, we have just published our 2022 research report, and when you leave this building, you will find it sort of on tables outside. You're invited to take a copy for yourself. This was a particular, 22 was an important year. We are sort of taking into account also the adjunct principal investigators, and their laboratories is 300 people or more, 50 nations. I think the one thing that probably stood out in 2022 was that Christoph Bock, who is sitting here, actually got the Erwin Schrödinger Award, which is the biggest award that the Academy is given to, and his son is here to see that, and of course his wife. So we are also very proud to have sort of accepted the challenge of including our heroes and the pioneers, the giants on whose shoulders we try to do our research, and they're represented here in a remake of the Beatles' Sgt. Pepper cover, where we have identified some of the person that each individual laboratory found inspiring. And I'm not going to tell you much more, so that you're curious and pick up your copy on your way out. Then very briefly, what is the Denise Barlow Award? Denise Barlow was a fantastic scientist, researcher that worked and was pioneer. She was the first female principal investigator at the IMP, and there are 4 institutions that she was associated with while she was in Vienna. And we have some of her pupils sitting here in the first row. And Denise was a very inspirational person, and when and the year after, or 2 years after, she went into pension, she died prematurely of cancer, and we felt heartbroken, and we are heartbroken because she was such an important member of our community, and we decided to make an award for her, and the award goes to somebody that during the PhD has contributed something that was mechanistically revealing. So it is rather sort of an activity that speaks to how things work at the molecular level. And these are the institutions that she's been associated with. And we are very, very happy that today we can give out the 4th such award. The first year, it was given to Matthias Muhar from Hannes Zuber's group. The second year, it was given, and still the same year to Julia Batke, who was in the Brennecke group. Last year, 2 years ago, we gave it to Annette Romanowska from Elvin Köhler's lab, and last year we went to Martin Jäger. Martin Jäger is here, and Martin Jäger, after this award, went to work with our lecturer, Benjamin Cravatt, in California. So you see that this is something that inspires the community and puts everything together. And so you will ask, who is this year? This year, awardee is Matthias Hintendorfer. And Matthias Hintendorfer, I would like to ask to come on stage. Please help me in applauding him. Alles klar. Wo ist das Ding? Maybe put it down here. Yeah. So Matthias, yeah, you can open it, and it's actually beautifully designed. The best part is that 5,000 euro that you can spend on helping the transition to postdoc. So show it to everybody. PhD students imitate Matthias and win a Barlow Award. That's the best that can happen to you. So, so Matthias has studied in Vienna. He was at BOKU. Then he went, no, okay, you were?
At the University of Applied Sciences.
Excellent. So, sorry. So he was at the University of Applied Sciences, and then I'm correct that you went to Hannes Tuber's lab to do your PhD. And Hannes is the one principal investigator in our community that has 2 winners of awards. So there's something special about him, and we are congratulating him along, and we'll ask him to come and take a picture at the end. But first I would like, okay, and now he's also close to the community because he decided to do a postdoc in Georg Quinter's lab, and Georg is sitting next to Hannes, congratulating to him as well. So maybe in a nutshell, you can tell us what your PhD was about.
Yeah, so I think the PhD started with a bunch of questions that are probably not unique to my PhD project, but probably were the beginning of 1,000 other PhD works. And that all of these questions resemble about the oncogenic transcription factor MYC. And as hopefully many of you know, MYC is super highly essential for the growth and proliferation of essentially all cell lines, of essentially all tumors. But the curious thing is it's rarely mutated itself, but it is always upregulated by some other factors. And so the question really was, how can so many different tumors from different tissue contexts, different driving oncogenes, all share the same feature of upregulating this one factor? And then so we decided to systematically address this question by setting up a genetic screen where we stain the MYC protein with antibodies to then be able to read out MYC expression level with a single cell resolution using a FACS assay. And then we combined this with a genetic screen where we essentially knock out all the genes in the genome to find which are the genes that drive this upregulation of MYC. And then the really curious thing was that we found the absolutely opposite of what we had actually looked for. So we wanted to find genes that if you knock them out, they lead to a loss and a downregulation of MYC. But we found something that if you knock it out, it leads to a drastic increase of MYC. And MYC accumulates and accumulates and accumulates until the cell dies. And so then long story short, the question we had, of course, what's going on? How does this work? And so we figured out that this gene that is called acrin-2 is responsible for bringing proteasomes into the nucleus. And so without acrin-2, the proteasomes are completely lost from the nucleus, and that's why MYC can't be turned over anymore, and then the cells die from that phenotype.
Fantastic, and I think sort of MYC, the name of this gene, is a cancer gene. Just for those of you, he said it, but in his amazing, successful way, he's very quick, and this is why he can get so much stuff done. But basically, we're talking about what makes a cancer cell, and the mechanism that regulates that process. Second question, what advice would you give to the current PhD students? What is it that you think you would like to tell them that they should do?
Yeah. So the one thing that I want to tell you is actually addressing not the PhD students, but the PIs. Yeah. No, so the one thing that really defined my PhD was the decision to not work on this set of questions alone, but to share everything in a complete like team effort, 50-50 with a second PhD student from start to finish. And that not only made it possible for us to answer these questions and get so much work done, but it also made the PhD a lot more fun to always have somebody to share your excitement with, to not only share the experiments, but just talk about everything you do and be excited and being able to show the results to somebody else, except of course for your PI. But that really made the difference for me, and I would recommend everybody to try to tackle questions in exactly that way.
Thank you. This is very interesting. And the last question that we have, well, it's the last question that we have. I mean, teamwork is what CeMM is all about. So, I mean, the last question that I have is, you're a rocket and you're on some trajectory. Where does the journey take you? What are your plans? What are your future plans?
I don't know, but maybe until 2 years ago or so, that thing scared me. I didn't know where I was going, and that was scary. But I think that has changed. I think I learned that I can be successful if I'm excited about something, and I can be excited about many things. So I think regardless of where the journey takes me, I think as long as I have fun, I'm going to be fine.
Bravo! And you have to move, so yeah, but you have to move because this is so. Make a nice face for Francie. Thank you. Okay, and now I beg the musicians. They will play two pieces. One is called Süden, and one is called Steja. And they will play them as if they are two parts of the same songs, but they really are two separate songs. And as I said, you can hear much more of that if you want, if you purchase their Tonträger, or if you are using them on Spotify or other means. So please. Bartolomei and Beatman! Okay, after this fantastic, energizing music, we come to the core of the matter and to the Landsteiner lecture. So let me very briefly tell you about this lecture. It started in 2007, and it was really meant to celebrate what you can call medical advancement in research. And as you know, it was sort of, the name is of Karl Landsteiner, we'll come to the next slide, is the most important name that this country has in terms of medical achievements. It was always here in this room, except for the COVID online and a brief interception at the House of Industry. And next year it will be still here. I tell this for my wife in case that she goes to CeMM like she did today for the lecture. So always. So Karl Landsteiner, as you know, has done this discovery that amazes us to this day, because just based on mixing blood, essentially, was able to find the concept of blood groups. And that mere fact saved, difficult to say how many lives, but we're talking about millions of lives that were saved. And of course, the history of this country and recognizing talents is mixed. And so he didn't get quite as much as recognition as he would have deserved, and much of the discoveries then were done also in the Netherlands, and most importantly in the US of A, a place that has taken up a lot of talents before and because of anti-Semitism, but not only, but then of course dramatically because of Nazism, but he immigrated before. I think that if we are thinking about sort of a person that stands as a hero, although heroes is a difficult concept, but as an inspiration, this is Karl Landsteiner, who never stopped to do great discoveries beyond this. So it was a person that, and as Austrians that are a little bit older will know that he also decorated the very important 1,000-shilling banknote that was the biggest banknote at that time before the euro set in. And now it's my great pleasure to try to summarize an incredible series of achievements of our lecturer, Benjamin Cravatt. First, I have to say, first very briefly, who were previous. I told Benjamin that Luke O'Neill came with his own band. So Bartholomew and Bittman, we had a lecturer that came with his own band from Ireland, and we danced to his music. So there is a precedence of this, but by and large, these were well-behaved individuals, and we are going back in time. The first one's John Kurian, and we've had a lot of fantastic discussions here. So now, and these are more pictures, and as I say, John Kurian was the very first one. But I also had some pictures of the same year when we had our first CeMM conference. It was called Drug in Action. I organized it with Ricky Eggert. You can see Benjamin unchanged, so you will easily recognize him here with his polo shirt, and here again. And here he's with Kai Jonsson and a few others, and Tijn Brummelkamp at the Natural History Museum. Here you see our current head of the Medical University in his youthful appearance before the management of stress. The stress of management hit him. Here is Ron Reyes together with Benjamin. And here I just put it up because you can see Terry Shepard, who was an important person in our community because he was the editor of the first journal dedicated on chemical biology, together with Nathaniel Gray, other sort of important people. So it was a great meeting, and thank you so much, Ben, for having followed us and supported us since. So very briefly about Benjamin. He's a professor and Norton B. Gilula Chair of Chemical Biology in the Department of Chemistry at the Scripps Research Institute in La Jolla, California, a fantastic and beautiful place. He is one of the persons that can rightfully say that he has contributed in making the field of chemical biology as important as it is. In fact, he has started whole branches of the chemical biology, most importantly, used activity probes, and he will tell you a little bit about that, to probe the proteome. What does it mean? It means that he was able to make chemistry that was interrogating not just the existence, but also the activity of proteins, of all the proteins at once in a cell, which, as you understand, is like you multiply the ability of testing one thing by testing at the same time thousands of things. And that, of course, gave you a much more complete picture, and out of it came beautiful discoveries. But he became very famous by previous work that work on endocannabinoids. So these are, as you will hear more in his talk, endogenous substances that are sort of important in the central nervous system. And he was interested in the metabolism of these compounds or these metabolites and generation. And based on this, and also from work that he was doing before, really the whole area of lipid metabolism in the central nervous system became a very favorite place to do his investigation, an area that is as hot as ever as we're trying to understand how neuroinflammation works and how to understand how pain works. And all of this are mainly based on molecules related to the molecules and to the type of molecules that Ben Cravatt is a world expert in. And I won't tell you all the great other things he's done because the introduction is already very much too long. But I also think that he's one of those people that, because of his approachable nature and his sense of humor and his being available, has been inspiring us very much. And I think that CeMM would not be the same without Benjamin Cravatt, which makes him a particularly welcome lecturer this year. And he also is a bio-entrepreneur. There are 3 companies that he has founded, ActiveX, Vividion, the last one, and in between, Abide Therapeutics. All 3 were sold, at least Vividion, we think, not at a loss. So he's an inspirational figure in that sense as well. You can be a very, very cool scientist taking no compromise on quality, be totally inspiring for your students and your postdocs, and make a lot of money doing this, which I think is, we've been trying to imitate him since many years. Dear Ben, I won't say much more about you. This community is really sort of all fans of yours. We are looking forward to your lecture, and then we will take some questions. So please come on stage.
I first wanted to offer my gratitude to Giulio for inviting me to participate in this really special event. As he mentioned, I have a deep fondness for CeMM. I feel like I kind of watched it grow from the very beginning, and it reminds me a lot of the early days of Scripps, which I've happened to have been at Scripps a long time. So, and I see some shared features. Lots of energy and enthusiasm for cutting-edge research, driven by innovative technologies, as Giulio mentioned, but also a very strong leader. And Scripps had Richard Lerner, who was my original advisor, and Giulio has a lot of features of Richard in him, in that he hires incredible people and supports them. Probably, I would argue, what they both share in common, which is atypical in the scientific community, is you can see that they actually derive more enthusiasm from the success of others than from their own success. And for those of you who know scientists, that's a rare feature, right? And it is a tribute to a great leader, so you're very fortunate here to have this wonderful individual and such a great institute. So before diving into the main topic of the lecture, I did want to spend the first slide with a message to the younger students in the audience, and that is the following. I'm very jealous that you have the opportunity to grow as scientists in this incredible era. This is really going to be the first era of research where we can honestly look at biology and say that we can use humans as our model organism for picking proteins and pathways for investigation. And this is largely due to incredible advances in DNA sequencing technologies that have allowed us to understand the genetic basis of human physiology and disease with incredible depth and breadth. And you can see this now in databases such as I've shown here, like the CeMM database, which is, I can do it this way, I forgot, this is really cool. Okay, the CeMM database, which is a database of Mendelian inherited disorders. So when I was a student 30 years ago, this was barely a database, right? It took labs 30 plus years to map the genetic basis of a single disease. Now, essentially, every Mendelian disorder has been mapped to its causative gene, and this gives us as biologists or chemical biologists a tremendous amount of material from which to derive hypotheses to test and study and ultimately treat human disease. Of course, with every major opportunity comes challenges. And what we are learning from the genetic basis of human disease is that it often points us to proteins and pathways that remain very poorly characterized. We know little about their biochemistry and their underlying functions, and in some cases may even be beyond the reach of druggability, which means we don't know how to make therapeutics to target those proteins. And that can lead to these sorts of diagrams that you'll see in review articles where the proteome, as Julia described it, which is the collection of all proteins in our cells, is divided up into a couple of different portions. One is a static portion, which contains protein classes that we know how to make drugs against, okay? That amounts to a small fraction of the proteome. And you have this ever-expanding portion of the proteome that has human biology disease relevance, and a relatively small overlap of those two groups. And so our lab, and many others around the world, are interested in trying to develop new technologies to bring these human biology relevant proteins into the realm of addressability through small molecule chemistry. And that's going to be one of the themes today of how we have tried to achieve that through an atypical type of chemistry that Julia and I together decided to call persistent chemistry today. And we'll discuss what that means in a moment. So first, though, I'll introduce this activity-based proteomics technology that Giulio mentioned and how we can use it to discover small molecule probes and ultimately drugs that bind proteins through studying them directly in living systems, okay? And we'll discuss why that is important. And then we'll spend the majority of the lecture on how activity-based proteomics helps to create a resurgent interest in what is actually a historically rich field of persistent chemistry, and how I hope to convince you and that we've gone from a field that chemistry was viewed as accidental and tolerated to a place now where it is intentional and celebrated. Okay, so if you were to ask how most small molecule drugs have been discovered over the last 20 or 30 years, it might look something like this. You have a human cell with a bunch of proteins in it, and you deem that one of them is important for a disease. And the first thing you do then is take that protein and take it out of the cell, okay? And you put it in a test tube so you can study it biochemically, okay? And then you may expose that protein to candidate small molecules that could ultimately, hopefully, within that pool, a molecule would emerge that would perturb that protein's function. And that, of course, requires you to develop a whole variety of specific assays for each protein that you would like to study, because they all perform different types of functions. So you need to have a specific assay for each protein, and this is where you start encountering one of your first problems with this traditional approach, which is that many of the proteins that are disease relevant, we don't understand what they do, so we don't even have assays to study them. Okay, so those already are moved off of the plate from being addressable with this format. But there are other features of this classical approach that left us and others concerned about its capacity to fully address our ability to create chemical probes and drugs against human biology proteins. One is that the efforts to put together these specialized assays are quite challenging and cumbersome. In fact, we have incredible scientists in the audience here today, like our lab, that have spent years developing assays against individual proteins. And that is important and laudable. But to do that across thousands and thousands of proteins, as Gilula mentioned, is extremely challenging, which means that most small molecules that are studied in this way, we don't really know how selective they are across the thousands of proteins that exist in our cell, because we had no way to assay those interactions. Other concerns are that when you pull the protein out of the cell, you are actually changing, in some ways, its biology, because proteins in our cells are modified by a variety of different dynamic processes, and only a subset of those will be retained when a protein is put in a test tube. So that could mean that you were actually studying the wrong protein form in the test tube for developing chemical probes and drugs that relate to its function in the cell. And finally, almost all of our proteins actually function in a specific neighborhood associating with other proteins in the cell. By isolating a protein away from its neighborhood, you also may change its biology and the effects it may have interacting with small molecules. So we were trying to think of ways that could address these challenges in a systematic manner. And this was the origins of this activity-based proteomic technology, which is going to take the principles of medicinal chemistry and apply them inside the cell. So we're going to study how small molecules interact with proteins in their native environment rather than in a test tube. And this amounts to adding now compounds to these cells, and then the compounds will interact with proteins in their native environment. And then, as Gilula mentioned, the key here is this activity-based probe, which are types of chemistries that can go in and interact with hundreds, if not thousands, of proteins in parallel, okay? Reading out changes in their activity such that if one of the proteins that interacts with this probe happens to have been bound by a small molecule in your library, it will block the probe interaction, and then you can read that out. So this allows you to essentially have a universal way within a cell biological system to identify how compounds interact with proteins. And this largely addresses the questions that we raised on the previous slide. Now you have a uniform assay, right? You don't have to develop a specific assay for each protein. You have an assay that can read out, as I'll show you later, thousands of sites and thousands of proteins in parallel for interacting with small molecules. And you're also assaying those proteins in their native state. So whatever modifications the cell's putting on them, we're accounting for that. And we're also accounting for their neighborhood because they're associated with their appropriate protein partners. So that is the origins of the activity profiling technology. Its goal is to understand how proteins function and how small molecules interact with proteins in a living system. Okay, so with that brief introduction, let's move on to how activity-based proteomics began to have an influence or an impact on a specific form of chemical probe and drug development that we call persistent chemistry. So this will start with our historical interests, as was mentioned earlier, in a system called the endocannabinoid pathway, okay? And that name endocannabinoid implies there might be exocannabinoids, and there are, the most well-studied of which is this molecule here, which is tetrahydrocannabinol, or THC. This is the psychoactive ingredient of cannabis, okay? So it's been known for millennia, well before the pharmaceutical industry existed, that cannabis produces useful pharmaceutical properties, including, for instance, reductions in pain sensation, and even improvements in neuropsychiatric states. But of course, these beneficial effects of cannabis come with a variety of side effects, including impairments in motor function and cognitive function, which limits the general utility of direct cannabinoids as therapeutics. These compounds act by binding a single protein in your brain, a single G protein coupled receptor called cannabinoid receptor 1. In fact, there's historically a lot of debate about whether these compounds acted through a single receptor because of the pleiotropic mechanisms that they displayed on the nervous system. But of course, it was the genetic knockout mouse, a wonderful technology that laid those debates to rest, because if you delete this receptor from a mouse, you can give it as much cannabis as you want. It shows no response whatsoever. And so, kind of a depressed mouse too, to be honest. But it ends up that, not surprisingly, this receptor did not evolve to recognize cannabis. They're endogenous ligands, as Gilula mentioned. These are bioactive lipids that represent neutral fatty acid derivatives, we called by these names anandamide and 2-arachidonoglycerols. So these are lipids that are made in your brain that bind and activate this receptor in much the same way that THC does. And so we and others hypothesized that if one could identify the enzymes that regulate these endogenous cannabinoids, maybe block the enzymes that degrade them, elevate the levels of those bioactive lipids, you could produce perhaps a beneficial subset of the behavioral effects you observe with direct cannabinoid receptor agonists. And one such enzyme that our lab helped to characterize is called fatty acid amide hydrolase, or FAH. This is the enzyme that will terminate the anandamide signaling function, hydrolyzing it to an acid. That acid does not bind the cannabinoid receptor, so this is essentially a signal termination event. This is the structure of FAH that we solved with Ray Stevens' lab. It's an integral membrane protein, so it binds within the membrane, and you can see the active site kind of faces the membrane where it's probably extracting out these lipids like anandamide to hydrolyze them. So as we began to this is mid-2000s, we began to really understand how this enzyme functioned. We got a call from Pfizer Pharmaceuticals, who were interested in trying to develop FAH inhibitors for neurological disorders. And so we set up an incredible collaboration with Pfizer scientists, including Kay On, Doug Johnson, and others, to the goal of finding small molecule inhibitors that would block FAH, elevate endocannabinoid signaling for neurological diseases. And at this point in time, the activity-based proteomic technology was just coming into being. So we did a traditional drug discovery exercise, as I mentioned earlier. We isolated FAH in a test tube, screened against a large compound library, and actually did find some incredible compounds that inhibited the enzyme, and one of them is shown here. And so this seemed like we were off and running towards a drug candidate. And then I got a call one day from Kay, a close friend and colleague at Pfizer, and she said, "We're in trouble." And I said, "Why are we in trouble?" She said, "Well, we've been mechanistically characterizing this compound, and we've even solved the structure, and this is what it looks like." I said, "That looks great. Molecule's in the pocket where it should be. It's right in the active site. What's wrong?" And she said, "Well, focus here." And what that was a physical bond between the small molecule and the protein occurring right at a key residue in the active site. So that's called a covalent bond, or as we'll describe it today, persistent chemistry. So that means these compounds will bind the enzyme and never let go, okay? It's an irreversible reaction. And I was kind of naive at that point. I was like, "Wow, that's cool. That's a neat way to inhibit a protein." And they were like, no, not cool, okay? Not cool, because at that point in time, Pfizer and other pharmaceutical companies were not really in the business of making drugs that permanently bound to proteins, okay? And we'll discuss why they were concerned about that in a moment. But let's first look at how most drugs bind to proteins, okay? They bind in a reversible manner, which means this is a small molecule. It will find a deep pocket on a protein, nestle in there, and bind, but not permanently. So it can, and this is how drugs like Lipitor and Januvia work, okay? So many drugs that thousands of, if not millions of people take operate by this mechanism, okay? This is a fantastic way to make drugs, but one of the reasons there are so many proteins in our bodies for which we do not have small molecule drugs against is because this type of chemistry really depends on having this beautifully shaped pocket that the reversible molecule can bind tightly to. And many of the proteins in our body look like this, okay? They don't have deep pockets for reversibly binding small molecules, which means these compounds come in, as we screen them, they kind of bounce around, and then they leave, okay? And we're left with no tool compound, probe, or even a drug candidate. And we actually surmised that FAH might be in this category because its active site was facing the membrane in such an atypical way. This might be very difficult for reversible small molecules to find access to its binding pocket. And so we thought this may be why the first compounds that were discovered operated by this persistent covalent mechanism. So the reason why we were perplexed initially about the trepidation that Pfizer showed for this pathway is that there are actually some incredibly useful drugs that operate by persistent chemistry, and you are well aware of many of them. So these drugs enter the protein, they bind, they form this permanent bond. That is exactly how drugs like penicillin and aspirin work. Okay, these are classic drugs taken by millions of people over, if not billions of people over the years. So why is the pharmaceutical industry so worried about this type of mechanism? Well, first off, in almost every case, it was an accidental discovery, okay? And this is never very scientifically satisfying, right? These were not purposefully developed as covalent drugs. They were discovered through phenotypic screening like penicillin, and then later on determined to operate by this persistent covalent mechanism. But the second reason why companies like Pfizer were concerned about this type of mechanism related to the concern that if you could permanently bind to one protein in our cells, how do you know you're not permanently binding to 10, 100, 1,000, right? And that would lead to concerns of safety, okay? And this is where we began to think that some of the activity-based proteomics technologies in our lab might be able to address this concern. So we first went back to Pfizer and Kay and said, you know, we really believe that this type of covalent drug mechanism, this persistent drug mechanism, may be ideal for a protein like Fa because you can bind this unusual, atypical small molecule binding pocket to form this covalent product that remains permanently bound. And they said, yeah, we agree with you. We agree that that may be true, but we need to have some evidence that we can do so specifically and selectively and not through binding this protein permanently while we bind thousands of other proteins permanently. We said, well, why can't we return to these very same assays I introduced to you earlier, where now we have these activity probes that are reacting with hundreds or thousands of proteins in our cells and ask, okay, if we take that Pfizer Fa inhibitor and add it to a cell, how many proteins does it perturb in this activity-based assay? So here's what some of the first data looked like. We took this compound, added it, well, actually treated animals with it across a dose range, and then ran an activity probe experiment where every band on this gel is a protein that reacts with an activity probe. And that reaction will only disappear if the protein has been bound permanently by the small molecule. And you can see across these dozens and dozens of proteins that are in the mouse brain, only one protein is blocked across the entire concentration range, and that is Fa. So we went to Pfizer with this and said, well, what do you think? They said, yeah, that's great, but you're only looking at dozens or hundreds of proteins here, and there are 20,000 proteins potentially in the human proteome, so that's not going to be good enough for us to try to progress this mechanism. And I said, okay, well, we can do actually one better, because once we have this type of advanced permanent probe, or a permanent inhibitor, we can convert it into an activity probe itself. And this actually uses and leverages some of the chemistry called click chemistry that, for those of you who follow the Nobel Prizes will know, this was the chemistry for which Barry Sharpless, the term that Barry Sharpless coined, and the chemistry for which he and Meldal and Carolyn Bertozzi received the Nobel Prize in 2022. So in this format, we take that Pfizer inhibitor, and now we just do a simple bond replacement, putting this tiny little alkyne on the compound, and that will allow us to trace any protein that it reacts with in any biological system, okay, in a permanent way. So then we take this alkyne variant, we do the same experiment, we add it to animals, and we ask, okay, across the entire mouse brain proteome, thousands and thousands of proteins, what does this compound react with? Here's what it looks like. One protein across the entire proteome, and when you look at the Fa knockout mouse, which is genetically needed for Fa, that protein goes away, okay? So this was actually the data that convinced Pfizer it was okay to take these compounds forward into clinical development. And there they have shown some very interesting, this compound has shown some very interesting clinical activity. I highlight one paper for you here that came out a few years ago where the study was designed to look at how raising the endogenous cannabinoid might affect cannabis withdrawal symptoms in humans. So, many of you may want to hear this, but it ends up that as cannabis has been used more widely in society, it also is beginning to present with a number of subjects who have abuse or dependence on the drug. And withdrawal to cannabis is subjects experience a variety of behavioral problems, one of which is sleep impairments. And so if you look at this study, what really was going on, it appears, is if they are treated with this blocker of FAW, the endogenous cannabinoids can improve their sleep quality while they're undergoing withdrawal from the drug cannabis. That leads to less return to cannabis use. Okay, so again, benefiting them in a neuropsychiatric way through sleep quality and other behaviors. So this was quite exciting to us, suggesting a positive neuropsychiatric effect of the drug. But right around that time, we were met with yet another challenge that I would call is beyond science. And that was that Pfizer decided to exit neuroscience altogether as an indication. Okay, and we could spend hours at the cocktail party describing why this is. I won't go into it in detail. I'll just say it wasn't because there wasn't need in neuroscience for new drugs, just a very challenging area of drug discovery. But I will give Pfizer credit that they didn't take their neuroscience assets and just put them on a shelf in the basement of their buildings. They instead out-licensed those drugs. And this compound, therefore, was out-licensed to Jazz Pharmaceuticals, which is now testing it in a phase 2 trial in post-traumatic stress disorder of huge unmet clinical need. And this initial trial should read out sometime at the end of this year or early next year. So the only thing that changed here is the name of the drug, right? So it went from the Pfizer name to the Jazz name. So if you want to follow this going forward, you have to follow this JZP-150 name now, because that's the new name for the drug. But this was an interesting example, right, where we were able to use activity-based proteomics to take a persistent chemistry mechanism and convert it into what is arguably the most potent, I would say it's the most potent persistent drug that's ever gone into humans and has had no safety issues whatsoever. So we were motivated by that to think more broadly about how we could leverage activity-based proteomics and other methods to explore the full potential of persistent chemistry for a chemical probe and drug development. But we also recognized we had to be honest with ourselves at that time. Because if you ask why we were so successful at creating an incredibly potent and selective covalent or persistent drug for FAW, the honest answer is we cheated. And we cheated because the residue that that drug was reacting with was a specific, unusual type of residue in our proteins that we refer to as hyperreactive, right? So the enzyme class that FAW belongs to uses a specific residue to perform its chemistry that is hyperreactive. In fact, this is the same way that penicillin works as well. Same residue, essentially, this hyperactivated serine residue. And that allows the medicinal chemist to come up with a whole variety of permanent chemistries, right? That will react with that hyperreactive catalytic residue and not touch any other residues in our proteins, okay? So that approach can be applied to a couple hundred proteins in our cells. That actually became Abide Therapeutics, the company that Julio mentioned, to pursue that objective. But there are 20,000 proteins or more in our proteomes, right? And this is therefore only applying to maybe 1% of those proteins. So if one wants to extend persistent covalent chemistry beyond the active site that bears a hyperreactive residue, one is going to have to think a bit differently. And it ends up that if you look across our proteins, there's another amino acid called cysteine, which is intrinsically quite reactive no matter where it exists in our proteins. It doesn't have to be within an active site and rigged. It is actually intrinsically quite reactive. And so this suggested that one of the directions persistent chemistry could go is targeting cysteine residues. And indeed, around the time we were putting the FAW inhibitor into the clinic, Pfizer, a parallel set of studies were being done by this person, Zhengying Pan, at that point in time, a scientist at a company called Celera in the Bay Area, who had discovered and developed this compound called ibrutinib, or Imbruvica, which is now a frontline therapy for treating B-cell cancers. So Celera had absolutely zero interest in developing persistent drugs at that point. So Pan was kind of doing this under the radar, actually initially making them largely as activity probes, right, to study the kinase BTK to which they react and inhibit, and how BTK functioned. But a year or two later, Celera actually largely got acquired by a company called Pharmacyclics. This compound was sold for $2 million, okay, as part of a package of other compounds like histone deacetylase inhibitors, okay? And then Pharmacyclics kind of looked underneath the hood and said, wow, we actually might have something important here, because they began to appreciate the role that the kinase that was targeted by this drug plays in B-cell cancers. And so they then developed that exact compound into a therapeutic, okay? And about 10 years later, they were acquired by AbbVie for $20 billion, okay? So a $2 million purchase became a $20 billion acquisition. This fascinating story is covered in great detail by this recent book that just came out from Nathan Vardy called For Blood and Money. If you're interested in any of the sort of interesting concepts behind biotechnology company development and the idiosyncratic nature in which compounds are developed into transformative therapeutics, I highly recommend this book. Okay, but I would also say that this story was also cheating. Okay, so this was cheat code, what I would call number 2. So what was this cheat code? So what Pan was doing here was taking advantage of a tight binding reversible inhibitor, okay, that bound beautifully in the BTK active site. That's the blue part of the small molecule you can see there, okay? And there happened to be a cysteine nearby, and so therefore he appended this reactive group shown here to react with that cysteine, okay? So that's essentially retrofitting persistent chemistry on a tight binding reversible scaffold. But I already told you that most proteins will not bend the knee to tight binding reversible ligands, right? So this is really another cheat code, okay? And for those reasons, at around this time, which is about a decade ago, there were sort of 2 lines of thought emerging in the community, okay? If you went to the pharmaceutical industries, they would still say, covalent persistent drug development is idiosyncratic, it's accidental, and it's a lucky break, okay? And we have no real capacity to believe that this could be done systematically and purposefully. But within the academic community, our lab and a few other groups saw the state of science differently. We thought it might be possible to develop, more generally, persistent chemical probes and drug candidates broadly across the proteome. And so I was trying to think about a way for maybe the audience to appreciate this dichotomy of views at the time. And I turned to this incredible lyric from Taylor Swift. I'm a huge Swiftie, so I'll admit. Where she says, I know my love should be celebrated, but you tolerate it. And I felt like at the time that was kind of what was going on. Those of us who loved persistent chemistry were watching others tolerate it, not celebrate it. Okay? So how are we going to solve that problem? This really meant in our minds that we had to come up with a way to discover persistent chemistries with no cheat codes, okay? It can't be because we're targeting some rare hyperreactive residue in a handful of active sites, and it can't be because we're leveraging reversible chemistry and making it persistent, right? Those are just narrow, interesting and valuable, but narrow approaches. And this was work that began with Ranthi Weerapana, Chu Wang in our lab, and then taken forward by Carrie Bacchus and Katja Vinogradova, where they systematically began to develop a platform that could look at how persistent compounds reacted with any cysteine in our proteomes, in our cells. So this platform would involve taking small molecules that are predisposed to react with cysteines, and you add them to cells, and they go in and they bind a pocket. If a cysteine is nearby, the persistent bond ensues. And the key is then to chase that reaction with a compound that will react with thousands and thousands of cysteines, okay? So it will read out thousands of cysteines across thousands of proteins, except those that have been pre-modified by these persistent chemistries. They essentially punch holes in that large profile. And you can read those holes out by a technology called mass spectrometry. This allows you to look at all these cysteines in parallel and determine which ones were physically bound by these electrophilic or reactive small molecules, okay? And this type of technology, when we first began to apply it, really began to shed light on the power of persistent chemistry. This is just a summary of some data that Carrie generated several years ago, where she identified, just using a handful, 20 or 30 persistent compounds, sites of persistent engagement on hundreds and hundreds of proteins, okay? And when you looked at those proteins in more detail, what was quite exciting is for the vast majority of them, this was their first evidence of interacting with small molecules, okay? So that was by the presence or absence in public databases like DrugBank. So this is kind of an interesting pie chart. If you look at this small sliver of the pie, where we're rediscovering evidence of drugability for proteins, okay? That means that our persistent small molecules are binding a pocket that we already know can bind chemistry. Most of these are protein classes like enzymes. And this makes great sense to the biochemist or the chemist because we know how to make reversible binding small molecules for enzymes. Okay, so now we're just finding permanent binding small molecules like I showed you earlier for BTK. That's not so surprising. What gets more exciting is when you look at the much larger slice of the pie, where this is now the first evidence of small molecules binding to proteins. And you see expanded representation of protein classes like transcription factors, adapter proteins. These are proteins that we have largely viewed historically as being beyond the reach of small molecules, suggesting that if one could unite molecular recognition with persistent chemistry, you could breach the traditional boundaries of what we consider druggable in the cell. Okay? And this was, I think, really the types of science that began to motivate a much larger and more purposeful investigation of persistent chemistry for chemical probe and drug development. And this was a company that Julio mentioned briefly, Vividion Therapeutics, that we started that pursued this goal and is now operating as a subsidiary of Bayer Pharmaceuticals. So I wanted to spend the last few minutes on one more recent story where this will get a little bit more deep into the nitty-gritties of the science, where I wanted to share with you how we've collaborated with a pharmaceutical company to use persistent chemistry and activity-based proteomics to discover a novel way to regulate an oncogenic protein in prostate cancer, and that is the androgen receptors. This is a collaboration with Janssen scientists, Sungju Koo, Kei On, Brahma Ghosh, and others. So most of you may be aware that the androgen receptor is a nuclear receptor that is a driving oncogene in prostate cancer. Okay, this is the major driver of this disease, and it has therefore been the focal point of pharmaceutical drug development in both academia and industry. I highlight this compound for you here, enzalutamide, which is a receptor as an antagonist of this androgen receptor. This compound was actually developed in academics by Mike Jung's lab at UCLA and is now sold as a therapeutic to treat prostate cancer. These drugs work fantastically well. They suppress the disease for many, many years in most cases. But ultimately, like with most cancers, resistance mechanisms will emerge. And the resistance mechanisms to androgen receptor antagonists are fascinating, including massively overexpressing the receptor such that there isn't enough drug on board to block it any longer, or creating proteoform variants, splice variants that lack the ligand binding domain but still preserve some capacity to act like an oncogene or a cancer-driving protein. And finally, in the most subversive way, the cancer will actually mutate the ligand binding pocket that binds these antagonists and convert them into agonists, which means now they drive the cancer rather than suppress it. And so this has led companies like Janssen to want to come up with new ways to suppress the androgen receptor in prostate cancer. And they developed an assay to look at how compounds could do so at the level of the transcript, the mRNA, the messenger RNA that encodes the androgen receptor. And from that, we screened a library of persistent compounds from our lab and identified one that could suppress the androgen receptor at the mRNA level. And over time, you would also see it suppress the receptor protein. But the mRNA went down first, which told us that whatever mechanism this compound was acting at, it was at controlling the transcript that encoded the androgen receptor. Here we were very fortunate that what we refer to as the structure-activity relationship for this effect was exquisite, meaning that we could make very small changes to the compound that would completely inactivate its ability to suppress the androgen receptor. Perhaps most importantly shown here by comparing this active and inactive pair of compounds, which by your quick eye might look totally identical. And they actually are physicochemically identical. They are enantiomers of one another, which means they only differ in their handedness or chirality, okay? One is a left-handed molecule, the other one is a right-handed molecule, okay? But only one suppresses the androgen receptor. The other has no activity whatsoever. And that's because our proteins are also chiral and will often only recognize one hand of a small molecule and not the other. Why am I going through this with you? Well, because for us to identify the target, the protein that was responsible for this bioactivity that's binding these compounds, this type of shared feature is incredibly valuable, because now we can use our activity-based proteomic experiment and ask what proteins in the cell react with the active hand but not the inactive hand, right? So when exquisitely similar chemical features, only differing in chirality, and what protein will discriminate in that regard. And there was only one protein that we discovered from activity-based proteomics that interacted with these compounds in that manner. And that was a protein that at that point I had never heard of. It was called NONO. What is NONO? NONO is a member, as I learned from reading the literature, a member of a clan of 3 sequence-related proteins that form homo and hetero oligomers and regulate virtually every step of mRNA maturation and processing, okay? And interestingly, if you look at the proteins that the family that NONO belongs to, only NONO possesses the cysteine that these compounds react with to suppress the androgen receptor. The other sister proteins, or we call paralogs, do not have that cysteine. And that'll be really important for understanding the feature of the mechanism that I'll describe in a moment. So we thought this was going to be really simple, right? We discovered a compound through a phenotypic screen that suppressed the mRNA that encodes the androgen receptor, okay? And the main target that looked like that compound bound to was a protein that regulates mRNAs, right? So it all seemed like it was going to work out quite well and very straightforward. And then Stefan Kathman, the postdoc in the lab who did this work, did what anyone would do at this point, like the CeMM researchers like Georg's lab and others. He used CRISPR-Cas9 technologies to knock out the NONO protein. The hypothesis being that these compounds were inhibiting this protein, and if we genetically knocked it out, we should get the same phenotype, suppressing the androgen receptor. And we got no such thing. When you genetically disrupt NONO, the androgen receptor remains exactly as it does in a wild-type cell. Both the full-length androgen receptor and these splice variants that I mentioned are the same in a wild-type cell and a cell that lacks this NONO protein that represents the target of our compounds. So we're scratching our heads saying, what is going on here? And then Stefan did another experiment. He added our active compounds to those cells and found that while genetically deleting NONO from cells did not replicate our compound's activity, it totally blocked our compound's activity. You can see that here. The active compound will suppress the androgen receptor in a wild-type cell. You get rid of NONO, the target that the compound binds to, it no longer affects the androgen receptor, okay? So this told us that our compounds were not acting as simple inhibitors, but were acting as another mechanism that required NONO to suppress the androgen receptor. And in fact, in a NONO knockout cell, the active enantiomer looks just like the inactive enantiomer and having no effect whatsoever. And I'm not going to take you through any of the excruciating efforts that went into working out this mechanism, but it's pretty fascinating. What ends up happening is this compound will trap NONO on RNAs. So it acts like almost like a glue, right? But a glue, instead of bringing two proteins together, brings protein together with RNA. And you can see that here visually, maybe only in the front, where when we treat cells with the active compounds that suppress the androgen receptor, they cause NONO to cluster into these nuclear foci, okay? These foci are thought to represent hubs of RNA-protein interactions, okay? So that tells us our compounds aren't simply knocking NONO off its RNA, they're trapping NONO on the RNAs to cause this aberrant mRNA processing, okay? So why is that interesting? Well, it's interesting because it's a case where a compound is producing an effect that is not mimicked by genetics, okay? And that means that if you look at the model for how this is happening, in a wild-type cell, NONO binds to its paralog proteins that I mentioned earlier, and all the sorts of mRNA processings occur normally. You get the normal mRNAs out, everything's fine. In a cell where NONO's been knocked out, why don't things go haywire? Because the paralog proteins take over and compensate. And they basically perform NONO's function for it in its absence. And in fact, just for the scientist geeks in the audience, the most highly upregulated proteins in cells that lack NONO are its paralog and sister proteins, okay? We have no idea how that's happening yet. That's an active line of pursuit in our lab. How is the cell sensing that NONO is gone and upregulating these sister proteins to overtake and drive its biology? But in a cell that's treated with the chemistry, the persistent chemistry that binds NONO, we've now trapped it, as I showed you earlier in that imaging, on RNAs. And so it doesn't matter if these paralog proteins go up. They can't displace NONO. It's still bound to these RNAs, and that's what leads to the suppression of the androgen receptor. So now we're trying to advance these ligands and optimize them further with the hypothesis that if we can make them selective enough, they could be useful as next-generation therapies for prostate cancer. Okay, so let me just finish with a couple final thoughts. These are interesting graphs. So this is a graph of showing you publications on covalent or persistent drugs over the years. And you can see for many years, there was nothing. And then in the early to mid-90s, some papers began to sprinkle in, okay? And it was around the time that I mentioned earlier where activity-based proteomics, the discovery of ibrutinib, that you saw this massive acceleration in publications in this field, right? And I think this field is now a very healthy, purposeful field aimed at, even in the drug discovery industry, at developing covalent drugs. And that is supported by this graph on the right, where you can see now there are dozens of covalent drugs that have been approved in the last decade or so. And if you look at how these drugs are developed, you'll see in most cases, if you look at the literature, behind their optimization is an activity-based proteomics experiment that says you are targeting the target that you want to treat the disease and not the thousands of other proteins in the proteome, okay? And so this is where, as Julia was mentioning earlier, academic labs, I think, can build technology platforms that support the drug discovery industry. It just may take them some time to appreciate and accept those technologies as being robust enough to develop a new type I should say more accurately, a renewed type of persistent chemistry-based drug development. Okay, so finally, again, speaking back to the students in the audience, and maybe everyone as well, especially those who might not be scientists, where are we at today? I think today, with our understanding of the genetic basis of human disease, it's actually incredibly common, much more so than it was 10 years ago, that if you or a loved one is ill, the physician that they're seeing will be able to tell them in incredible detail what is mechanistically wrong with them, okay? But I would actually say that we're not much better at doing anything about it, okay? And I think that's kind of a sad statement, right? Because I don't think that most patients who suffer from a disease care too much about the mechanism if we can't actually still treat the disease, okay? So that is, in my mind, this century's challenge, okay? To move from explaining what is wrong with someone to finding a way to treat and rectify that disorder. And I believe that persistent chemistry will be one of many types of modalities, drug modalities, that can be used to address that challenge. What do I worry about? Well, as in most fields, pendulums tend to swing violently in one direction or another. So while we started out in persistent chemistry with no one in the pharmaceutical industry wanting to listen to us at all about its potential to serve as a new therapeutic modality, we're now entirely on the other side of the spectrum, where covalent chemistry and persistent chemistry is being applied across the industry, and in many cases without the rigor that we and others believe is required to make sure those drugs truly do get optimized to be safe in humans. And I will call attention to one example where this led to a tragedy. This was a persistent drug developed by Bial that maybe some of you had heard about a few years ago, went into humans, caused rare neurological disorders in patients, some of which were permanent and even led to a death in one case. And so this is an example, I think, where a persistent drug was not evaluated by the types of technologies we described today to optimize its selectivity. I will say that I am encouraged by the fact that major drug agencies are beginning to appreciate the importance of this type of rigor. Some of you may have seen this, but the FDA recently came out with something called Project Optimus, which is an effort, at least in cancer, to try to find a way to enforce a distinct way of drug development. So if a company today develops a new cancer drug, even if it's targeted against a single protein in your body, and they have ways to measure that targeting, they will still take that drug and dose it in humans all the way up until its maximum tolerated dose, even if that dose is 100 to 1,000-fold above what is needed to block that target. That makes no sense whatsoever, okay? And it's just traditionally how drug development was done before you could measure with technologies like the ones I described today, how a drug is binding to a specific protein. But we have those technologies now. And so the FDA is stepping in and saying, no, we're going to mandate that you figure out what the dose of the drug is that hits the target and don't go above that. Because going above that doesn't do anything but bad things, right? It can only hurt the patient, not help them. And this is especially important as we combine therapies together where they need to be particularly safe. So I think I'm optimistic that we will avoid excessive celebration here for this type of modality, but it's still worthwhile keeping that in mind, right? And especially for the students in the audience. Having been in this business for 30, 35 years, it's stunning to see how the pendulums swing back and forth, right? And there's violent confidence on either side of the pendulum when it exists there. And so just be sober and more circumspect when fields gain excessive exuberance, just as much as you may when they may have excessive pessimism. Okay? All right. I mentioned most of the lab members who worked on these projects along the way, but it's been an incredible pleasure to work with an innovative, energetic, persistent group of scientists across all disciplines in chemistry and biology. And none of our work would have been possible without incredible collaborations, not only with pharmaceutical companies like Pfizer and Janssen, but also academics like Stuart Schreiber and Jean Yao and others. And again, thank you so much for having an opportunity to share some of our work and my thoughts with you today. And if you have any questions, I'd be happy to answer them.
Fantastic. We would have a beautiful medal, except that it was engraved wrongly. So you will receive it by mail, but you get the other best of Vienna, which is a Sachertorte, which is a chocolate cake.
Excellent. Even better.
With a promise that you will get the real thing as soon as possible.
Thank you.
So thank you so much. We are taking questions, please. Varun, in the back.
Thank you very much. It was a fantastic talk. Given the success of the persistent chemistry on the protein that was tracked on the RNA, would you consider potentially targeting the RNA itself?
Oh,
with
wow,
the
what
persistent probe?
what a great question. So the question essentially is asking, and I should have maybe specified that the cysteine that the persistent chemistry binds to is nestled between the two RNA binding motifs. So your intuition that the compound may be projecting out towards the RNA substrates is valid. And that's actually what we kind of have to do if we want to convert that mechanism from a really cool chemical probe into a therapeutic. Because right now, we're trapping too many RNAs beyond the antigen receptor to make it, in my view, therapeutically useful. But that's exactly the direction we're going. We're asking if we move those molecules out of the no-no cysteine pocket into the space where the RNA is binding, can we achieve some structure-activity relationship that would involve RNA recognition that could refine or restrict the regulation of RNAs to those that we want, like antigen receptor, and avoid others that we may not want to block. So yeah, that's exactly where we're going right now, and it should be interesting. I mean, obviously with technologies like RNA sequencing, it's actually technically feasible to be able to answer that question, right? I don't know if we'll be successful, but we will be able to test the hypothesis, yeah.
More questions?
Yeah. Oh, good, sure, yeah.
Rob and then Georg.
Wonderful talk. You already sort of alluded to it, but does it mean that for persistent chemistry, on average, the dosage is lower because it's such a permanent thing?
That's a great, wow, what wonderful questions. Okay, so I'm gonna go on a little bit of a side note here, and I'll get back to your question. So I almost put a slide in about this, but I thought it would be too granular. But there was one other event that happened in that 2009 to 11 timeframe that had, in my mind, an outsized influence, but still a striking influence on the drug discovery industry. And that it was a meta-analysis done by, I think, a toxicology group initially at Pfizer, where they said, what are the features of toxic drugs? Okay, and I think all the most of the pharmaceutical industry thought, reactive metabolism, reactive pocket, exactly the things that I was mentioning would be the features of toxic drugs. No, there was only one feature that related in a statistically significant way to toxicity: dose. That's it. Doesn't matter if reversible, irreversible, persistent, not, dose. So you're 100% correct that the best way to avoid toxicity, whether it's persistent chemistry or not, is dose. And we're achieving that with variable success right now, okay? So I would not say, to answer your question, that as much as I would like to argue that persistent drugs are more likely to go into humans at a lower dose. That is true with the FAW drug I described. And that drug got almost too potent. It ends up in drug discovery if a drug goes below a milligram per dose per day, it's actually hard to formulate, right, and process. So you don't want your drug to be more potent than that generally. But like if you take the Amgen G12C KRAS inhibitor, right, that's a persistent drug. It's given at 1 gram a day, okay? And they've had tremendous difficulty combining it with other drugs, right? So it's relatively safe on its own. You start trying to combine it, you get all these idiosyncratic toxicities, and that is probably related to dose. So I think to answer your question, the aspiration is to get, the Pfizer review basically said anything below 20 milligrams a day, there's no evidence ever of a drug being given at 20 milligrams a day or below, giving idiosyncratic tox. So that's become sort of an aspiration. We don't always reach it, but that's the goal, yeah.
Thanks.
Georg.
Yeah, thanks for a fantastic lecture. As these covalent drugs stick on the protein for its lifetime, that also means once the protein gets chopped down into peptides, these peptides will be displayed and shown to the immune system, and there will be still that covalent moiety or persistent moiety on it. Yeah. And I think there are some aspirations to combine covalent chemistry with, medicines that would reprogram the immune system. So I was just curious to hear your thoughts on that line.
Yeah, so what Georg's pointing out is that, let's say there's a protein that exists inside the cell and you form a permanent bond with a small molecule. That protein goes through some sort of half-life and then gets degraded by something called the proteasome, which leads to little tiny peptides. But that peptide that has the drug bound to it, the drug will stay bound. And that may get presented on the surface of our cells in MHC complexes for T cells to recognize. And this has been a concern, right, that it might lead to idiosyncratic immune pathology. That hasn't really been proven to be the case yet, even with drugs like ibrutinib, which are, I grew up doing catalytic antibodies, so I kind of look at small molecules as haptens, right? I know what looks like a good antigen, and ibrutinib looks like a great antigen, right? And yet still not idiosyncratic. So then the second part of your question is, can you then, if it's not causing tox, can you leverage it for immunotherapy? And there are active efforts going on, as you may be aware of. The G12C KRAS field is one of them, where labs are trying to engineer even cellular therapies, like expressing antibodies or TCRs that have been engineered to recognize that peptide antigen, covalent antigen. The challenge, I think, is that it gets back to dose, but now the dose relates to the concentration of the peptide antigen on the surface of cells. It has to reach a certain level of concentration, or it's hard to get the immune system to react in a way that drives biology. And I think the fields of cellular engineering have encountered this as well, recognizing that the concentration of antigen is really an important factor. So I think for an abundant target, and maybe BTK is pretty abundant, that might be a very interesting study to see if you could actually get even more therapeutic benefit from targeting the peptide antigen of the permanent chemistry as well. Yeah, it's a good question.
If you move to this side, maybe we inspire the right. This part of the audience.
It's getting biased, I see. Okay, yeah.
Andreas.
Thank you. Wonderful talk. Maybe a question that's less creative, but you showed a lot for cysteine, and in the beginning you showed serine. What's your thoughts on amino acids that can be used as attachment point for drugs?
Yeah, so the question is, can we extend covalent or persistent chemistry beyond hyperactivated serines or cysteines? And I think the answer is yes in a qualified way. No amino acid in our proteins is as intrinsically nucleophilic as cysteine. And I think we have to acknowledge that, right? But there are other amino acids that are closer to being nucleophilic than serine, right? Tyrosine, lysine, even aspartate and glutamate. They're only a couple orders of magnitude away from being pretty reactive. And so they can be pKa perturbed, or a little scientific there, but they can have their reactivity perturbed to become addressable with covalent chemistry. That's actually, there's a drug on the market today developed by Global Blood Therapeutics that targets the N-terminal amine of hemoglobin, right? And through an aldehyde, through covalent chemistry. Now, that's like cheat code 3, right? Because the N-terminal amine has a pK of like 7, right? So it's basically way more nucleophilic than a lysine side chain. I think what'll be interesting, and I'm not sure about, is whether unperturbed amino acids like lysines or tyrosines can really be targeted with covalent or persistent chemistry specifically. My intuition is if it happens, it'll be more of a retrofitting mechanism where you have a tight binding reversible binder that creates an effective molarity in proximity to that residue, and then you can form the covalent bond through just residence time. What is the utility of that? Well, you know, if the protein had a really long half-life, you know, you could drive more pharmacodynamic activity by staying persistently bound through covalency. So there could be some value. But I'm a little skeptical that it'll be trivial to just jump to additional amino acids given the reduced intrinsic reactivity that they have. But many labs are trying, so, and I'd love to be proven wrong, so yeah.
I actually have a proposal. What if you think about the question you may have for Ben during the cocktail, and you join me not only in thanking him, but asking Bartholomew and Bittman to give an encore. It's not agreed. It's not agreed. We may have to pay you a little bit more, but I think it would be a great way. And then you can think about the question, then have that. So the magic word in German is called Zugabe. So you have to say Zugabe, 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 Zugabe. That alleviates and then at the mandola, the mother of the mandolin, I suppose, Clemens Bittmann, fantastic, and thank you also to Matthias Bartolomei. Bartolomei and Bittmann, thank you very much. And thank you to our speaker, Benjamin Cravatt, for a fantastic lecture that is inspiring. Now let's follow him downstairs where we can all drink and eat something to celebrate our community.